In a previous video, we looked at the structure of the nucleus found within a cell. As an extension of that, in this video, we are going to look at the contents of a nucleus, specifically the genetic material or DNA. The cell goes through different stages during cell division. Correspondingly, the nucleus also undergoes changes. A cell usually remains in its interphase stage. The genetic material within an interphase nucleus is loose and a network of fibers called as chromatin reticulum or simply the chromatin. But did you know a single human cell has approximately 2 meters long thread of this chromatin? Let me put into perspective what 2 meters means. This is the average height of a male and a female globally. So males it is somewhere around 170 centimeters and for female it is 145 centimeter. A surfboard is 2 meters in height. So is the javelin. A typical door frame is also 2 meters in height. My favorite basketball player, LeBron James, is also 2 meters. So that is roughly 6 feet 5 inches. Now imagine a thread which is 2 meters in length. What is even more bizarre is how this 2 meter long thread needs to be put into the nucleus that is roughly around 5 to 10 micrometers. This is almost like trying to stuff a fully grown elephant into a suitcase. Chromatin is not just DNA. They also have specialized proteins like histones. Histones are positively charged proteins that bind closely with a negatively charged DNA. We also have non-histone proteins as well as RNA. All these components help the chromatin to be wrapped up nicely within the nucleus. How it does that? We will see that in another video. The 2 meter long DNA within the nucleus is distributed among 46 chromosomes. In an electron microscope, this is how the chromatin reticulum looks like. See these bright areas around the nucleolus? The genetic material looks different in different stages of cell division. You can see chromatin reticulum over here, but at the same time here we have a different form of the genetic material. The chromatin slowly changes form just before cell division. In this video, there is no sign of chromatin at all. We see another form of the DNA altogether. And here too, the cell is undergoing cell division. These are some of the earliest electron micrographs of the chromosomal form of our DNA. Chromosomes are visible only in dividing cells, not otherwise. So the chromatin fiber condenses into a chromosome. But wait a minute. In the electron micrograph, we see 2 arms in a chromosome. So how is that possible? That is because just before cell division, the chromatin reticulum undergoes replication and forms another copy of itself, which then condenses into the other arm in a chromosome. These 2 arms are called as the sister chromatids of the chromosome. They are held together at a spot called as centromere. Centromere is found in a narrow portion of the chromosome called as the primary constriction. On either sides of the centromere, we have a disc-shaped protein called as kinetochore. Kinetochores are very important during cell division because they are the attachment sites of spindle fibers that help in splitting the genetic material. So we have our primary constriction. And along the chromosome, we also see another narrow region called as the secondary constrictions. These are usually non-staining because the DNA at these spots do not take in the staining dye. These secondary constrictions can appear anywhere on the chromosome, meaning its position is not fixed. The segment of DNA beyond the secondary constriction is called as the satellite fragment. And the chromosomes that show satellite fragments are called as the satellite chromosomes. So not all chromosomes have a satellite segment. So this is the primary constriction. Here we have our secondary constriction and the satellite fragment beyond it. It has been noticed that chromosomes can bend only at the primary constriction during cell division. The position of the centromere is not always in the middle of all the chromosomes. It's different, and based upon the position of the centromere, we have different morphology for chromosomes. If we have a chromosome where the centromere is almost in the middle of the chromosome, and therefore we have 2 equal arms. These are metacentric chromosomes. If the centromere is starting to move to one side such that we have one arm that is slightly shorter than the other, this is a submetacentric chromosome. As the centromere moves further towards one end, we have one side which is very, very short and another side which is comparatively long. These are acrocentric chromosomes. And finally, when the centromere moves to one end, these are called as terminal centromeres, and such chromosomes are called as telocentric chromosomes. This is the micrograph of all the chromosomes within a human cell. I'll give you some time, see if you can spot the different morphologies. So this chromosome right at the center is a metacentric chromosome. This chromosome over here is a submetacentric one. Up over here, we have an acrocentric chromosome. And towards the left on the bottom part here, it is a telocentric chromosome.